Welcome back to Miss B Biology. In this video, I will be showing you step by step how to go about testing a leaf for starch. So grab your book and grab your pen and let us begin. How can we prove that a plant has photosynthesized? Let us remind ourselves of the equation for photosynthesis. It is carbon dioxide plus water in the presence of light to produce glucose and oxygen. The glucose that is made by plant is used for respiration. It is also used to make cellulose for the cell wall of plants. Plants also store excess glucose as starch. A starch test can be carried out on a leaf. The presence of starch in the leaf is evidence that photosynthesis took place. These are the things that you will need to carry out the investigation. You need a leaf that is coming from a plant that was exposed to light for several hours. You need your tripod stand with Bunsen burner, a beaker with boiling water, test tube with ethanol, a white tile, and your iodine solution. The first thing you would do is to remove a leaf from a plant that was exposed to light for several hours. Next step is to boil the leaf. Why are we boiling the leaf? We're boiling the leaf to kill the leaf and end all chemical reactions in the leaf. The boiling of the leaf will also break down the cell wall and cell membrane so that the iodine solution can enter the cells and react with the starch. Next step is to remove the leaf from the boiling water and place it in a test tube with ethanol. Next step, place the test tube with ethanol and leaf in the beaker. Please ensure that the Bunsen burner is off as ethanol is highly flammable. Boiling the leaf in ethanol removes the chlorophyll from the leaf. But why are we doing that? The chlorophyll would mask the color change if iodine were to react with starch. Do you notice how green the ethanol is? And this is because the chlorophyll was removed from the leaf. So the next step is to remove the leaf from the ethanol. At this point, the leaf is very dry. We would then rinse the leaf in the water to soften the leaf. Next step is to place the leaf on a white tile and then add a few drops of iodine solution to the leaf. And here is the result. The leaf changes to a blue-black color when iodine reacts with starch. If the same investigation was done on a plant that was locked away in a dark cupboard for several hours, the color of the leaf would be brown or the same color of the iodine. This is because the plant would use up the stored starch in the dark, as the plant is not making any new glucose by photosynthesis. The plant did not photosynthesize as no light was present. Thank you so much for watching and all the best in your exams. Bye bye.